Hi class, this is your instructor, Skylar Huff, and welcome back to yet another lecture overview. We've now made it into the 9th chapter, and this is all about the muscular system. Prior to getting started, I will say this. Please ensure that you take the time to begin preparing for Lab Test 2. In preparation for Lab Test 2, you all will be given a list of muscles, and those muscles given will be used to be tested on. Typical of which is we would use models for the test. And of course, in this case, we will be using an online version of the test. In other words, the test will be administered via Canvas. In the event that it's not administered via Canvas, you'll be let know, of course, during lecture. In the meantime, I'll say it this way. The test will consist of two figures. Those figures are figure 9.23 and figure 9.24. Each of which can be found on page 314. Use those two figures as well as those labeled muscles. I'll give you all that refined list of muscles here shortly. Now let's get back to the issue at hand. This is chapter 9. So this is all about the muscular system. So as it's laid out, we'll begin, of course, with the structure of skeletal muscle contraction. We'll get to how muscle responses, and then end things thereafter with smooth muscle, cardiac muscle, very briefly, and how we change over time. And I don't mean evolve. So there are three types of muscle. Of course, you should know by now, having done tissues by now. They are skeletal muscle, smooth muscle, and cardiac muscle. So with that, skeletal muscle is attached to bone. It covers the skeleton. We have those long cylindrical fibers, which are, of course, maybe known as cells, but I'll call them long cylindrical fibers. It is striated, and with that, I'll say it's voluntary. That means you control it. I control it. It rapidly contracts. However, with that rapid contraction, there is, of course, I guess I'll say something that comes along with it, meaning you can't contract so rapidly and not have a drawback. The drawback is, of course, that skeletal muscle tires easily. So yes, it's quick to contract in the very same way. It, of course, easily tires. And skeletal muscle is tremendously powerful, yet adaptable. And when I say that skeletal muscle is quite adaptable, I'm saying that you can pick up this huge textbook we have. Maybe the textbook is not as huge as others that we've used in the past, but this textbook may be an approximate 3 pounds. So you can pick up this 3-pound textbook, and then in that very same hand, or even in your other hand, you can pick up, of course, the paperclip that weighs, of course, a mere ounce or a few ounces. How adaptable skeletal muscle is. Next up is smooth muscle. So smooth muscle is found in the walls of hollow visceral organs such as the respiratory passages, the bladder, and even the stomach. So what the smooth muscle does for us is it forces fluids through the internal body channels, and it's elongated, smooth cells. So those cells are smooth, such as I would say like a cigar, even though I don't smoke, and if you happen to smoke, I recommend you, well, if you like to quit smoking, there are ways to quit smoking. I just hope that you all don't smoke, but if you do smoke, you do smoke. Just know that smoking can cause cancer, So getting back to where I left off, smooth muscle is involuntary. That means we are not able to control it. And then, of course, I'll say it's sometimes referred to as being visceral smooth muscle because it's found class in our viscera, those internal organs, those organs class that we cannot control. And with that, I say contractions with smooth muscle, they're quite different than what we just saw with skeletal muscle. Contractions here, class, are slow and sustained. And as I say, smooth muscle can contract and remain in its contracted state and not tire. That is quite unlike what you all just learned about that known as skeletal muscle. Now back to cardiac muscle. Cardiac muscle can be found, class, in the walls of the heart, and it, like skeletal muscle, class, is striated. So it's striated, and then, of course, it is involuntary. So involuntarily, class, those contractions occur. And this occurs without nervous system stimulation. That's the way in which, of course, this known as cardiac muscle contracts. And as I've mentioned that, when it does contract, it contracts at a steady rate. And that steady rate at which it contracts is set by the heart's pacemaker. So although there are neural controls that can speed up the heart for a brief period, such as when you're running, the heart class is typically under the control of the vagus nerve, which of course allows us to remain at approximate 70 beats per minute. So let's get further into a couple of more, well, I'll say 4 further characteristics of muscle. So muscle, by definition, class, is excitable, or it exudes or exhibits responsiveness. So what I'm saying about it here is that it receives and responds to stimuli by changing its membrane potential. Now, if there was ever anything I would write down and read and understand, is the way in which muscle can change its membrane potential by responding to those stimuli. So in muscle, the stimulus is typically class A neurotransmitter, and it's going to be released by a nerve cell. In the case of skeletal muscle, that primary neurotransmitter class is acetylcholine, which you all get to when you get to your discussion for the chapter. Number 2 would be contractility. So muscle exhibits that ability to shorten forcibly upon adequate stimulation, setting muscle apart class from all other tissue types. The third class is that muscle exhibits extensibility, in that it has an ability to stretch. So yes, muscle can shorten while contracting, and it can also stretch when relaxed, even beyond resting length. Lastly, muscle exhibits elasticity. So that's when your muscle class is able to recoil and then resume its resting length upon stretching. And it does feel good to stretch. I just stretched. Up next class are 4 muscle functions. The first of which, class, is to produce movement. And I think we all know about that function of muscle. So by way of locomotion or even allowing manipulation, and it's because of skeletal muscle. Blood circulates throughout the body because of the rhythmic beating of our cardiac muscle, the heart. And smooth muscle in the walls of those blood vessels, it's there, in which, of course, also helps maintain blood pressure. And that does include, class, the digestive, the urinary, and even those reproductive systems. Smooth muscle is quite involved. The second of 4 is that muscle functions, class, to maintain posture and body position. So with that, that means that our skeletal muscles are there, allowing you to sit in that chair, or allowing you to, I guess, lay across that bed, or allowing you to just stand as you take notes, or listen to this lecture, or even as you run. Maybe you are running and listening to lecture again as you run. Endorphins, though. Next up, muscle functions to stabilize joints, and lastly, it functions, class, to generate heat. So when I say it that way, muscle, class, particularly skeletal muscle, generates copious amounts of heat. So with that happening, it allows us to maintain our normal body temperature, especially on those cold days. I remember after saying that, I remember those days when I would stand outside in the fall and, of course, into winter, back where I'm from in Mississippi, waiting on the school bus. And some of those days, it would be downright cold. But the very same way, me being as young as I was, I didn't want to wear those big coats that my mama and daddy wanted me to wear. So I might have had on something very thin. But of course, your body, by way of, I hope you know it, is it positive feedback or is it negative feedback? Yes, by way of negative feedback, class, your body will then cause you to shiver to, of course, get that body temperature up, getting you back to homeostasis. That's just one way. So now let's get back to where I left off in your textbook, class, on page 293. And we're now, class, at the structure of skeletal muscle. Let us begin. Skeletal muscle, as it is, it's structured, class, in a way that it's, well, I gave you a little bit about skeletal muscle, but the way that it is structured, and I'll say turn yourselves over to, since your text starts there, I guess I'll say we'll start here on page 294. 294. There we are. The way it's structured, class, is that you have those long cylindrical muscle fibers, sort of like, I guess you could say, seeing a number of columns really close together. Or even looking at those 4 fingers you have, meaning your small finger, a little finger, a pinky finger, the ring finger, the middle finger, and then your index finger. If you put those together, of course, you would then have 1, 2, 3, 4 muscle fibers. And at the periphery of those fingers is where you would have those nuclei. And yes, I did just use the word nuclei. And I'm saying that because structurally, skeletal muscle is wholly different from what you've learned about that typical cell back in maybe one of our first chapters, being I think it's called chapter 3, or even what you learned in your biology 1 course. So those muscle fibers are multinucleate. I say again, those muscle fibers are multinucleate, or you may even want to call those, yes, fiber refers to skeletal muscle cells. And it's because they have a fiber-like appearance. That's why I just use the example class with those 4 fingers. So they are multinucleated cells with nuclei that are right beneath the sarcolemma. And I haven't gotten quite there yet, but keep in mind, class, that the sarcolemma is just that cell membrane here, of course, in muscle. And that reminds me, class, on page 293, I said this a long time ago in the first chapter, and even likely in that second chapter, but please make sure you all review understanding words. As we continue on through the course, there are a number of words you may not have heard before, or words that may sound like this word or that word. And I just want to make sure that you all are able to take these words in and make sense of these words. I'll continue where I was. So with that, skeletal muscle has what is known as a sarcolemma, and that sarcolemma will be that cell membrane of this muscle. Then there's what is called the sarcoplasm. So the sarcoplasm would be, of course, the cytoplasm, that cytosol here in muscle cells. And with that, I don't think I'm going to go over many more terms at this moment, but I'll go over a few other things. So to add to that list of things that are called terms, the next term up, class, is known as the sarcomere. So the sarcomere is that functional unit. I repeat, that sarcomere is that functional unit of muscle contraction. So with the sarcomere, I wish I could draw this a lot better than what I'm seeing in my brain or on paper. But if you look closely on page 296, you can see the sarcomere. You can look at figure 9.5, B as in boy. 9.5, B as in boy. And when I refer to that sarcomere, I'm referring to those things that allow contraction to occur. And having said that, I'll go back to one thing, and this thing that is known as the myofibril. I'll go back to the myofibril. As I mentioned the myofibril, I just mentioned that myofibril as being, of course, those parallel structures within the muscle itself. So the myofibrils are what allows muscles to contract. So it's just those overlapping, as you see. Let's look at, I guess I'll go now to on page 296. As you see on page 296, look at figure 9.4. I guess I'll say 9.4a, and look to the right. You're seeing those thick and thin filaments. Well, those thick and thin filaments are what make up that myofibril. So as I say it that way, it's just those overlapping parallel groups of thick and thin filaments. And of course, that is what will eventually get to where I get to about muscle being striated. I haven't quite gotten there yet. So with all of these, I'm going to just go to the figure to do this with you. I'll stop here. And this is skeletal muscle structure in a nutshell. Let's begin. So as you look closely with me, I guess I'll begin here at the bottom. So here at the bottom, right there in the center, that is, I guess I'll say where I'll begin by describing it. And I'll start there as being the M line. That's M as in Michael. That line that's purple that goes up and down, that's the M line. M as in Michael. So flanking the M line on either side is going to be what is known, class, as the A band. A as in apple, the A band. And you can now see that A band here with that very same M line. So I'm doing a number of things here, and I'll go back in just a moment. So that M line, class, is what anchors those thick filaments. And I'm going to give those thick filaments another name since I'm here already. And you can call those thick filaments, class, myosin. You can call those myosin. You may hear me refer to them later on, not much later, but as being myosin heads. So even inside that A band, there's another structure, And that structure class is called the H zone. That's H as in hello, the H zone. So the H zone isn't necessarily labeled here, but in that H zone, that's just that very center of this A band. So now I'll go just outside of this A band where you see where it's a lot lighter. So just outside of the A band where it's a lot lighter, this class is known as the I band. I is in it. So the I band is wholly different in that it looks, I hope you can see that it looks lighter than what we just saw as that A band. So zigzagging, yes, I just said that, zigzagging in the center of the I band is what is known class as the Z line. And you may hear other textbooks call it a Z disc. But that Z line class is that point of attachment for those thin filaments. So now that you've gotten this much, I guess what I'll do now is saying that from M line, which there is one shown here, to M line, which there is not necessarily one shown there, would make up what is known class as the sarcomere. So it's those myofibrils that are essentially, of course, sarcomeres joined end to end. So what happens here is, as contraction occurs, you get this slide. And I did say slide, which reminds me of a song by, I suppose I'll say Drake. Let me stop. But as the fiber slides, it will slide class, of course, from what is in that I band here, and from what is in the I band there. So those will slide and slide and slide until, of course, the muscle fiber shortens. And we'll get farther into this sliding filament theory of muscle contraction. We're just not quite there yet. I'll do a few more things with structure before I go there now. So what I'll get to here is going to be those connective tissue coverings. And if it needs to be said a different way, this is what covers muscle fibers. And this class can be found on page 293, left-hand column. So it's layers of connective tissue that cover skeletal muscle. So that individual skeletal muscle is separated from adjacent muscles and held in position by these layers of dense connective tissue called fascia. I don't know if any of you all happen to, oh boy, how do I say this? I'll just say hunt. If ever you've gone hunting, and harvested an animal, you likely have seen that fascia if in fact you field dress that animal. So what I'm getting to here is that connective tissue that surrounds each muscle, and it may even project beyond the ends of the muscle to that cord-like tendon. And I don't know if you all happen to ever cook chicken, which I do cook chicken. Tonight was fish, however. The tendon class is there, and then you see that fascia here, which of course would be that connective tissue sheath or covering that covers the muscle. So continuing on, what we have here, class, is that those layers of connective tissue class that cover, I'll begin class externally. So the very outermost, or the outermost class, connective tissue layer, is known class as the epimysium. Epimysium. So that what class surrounds the individual bundle of muscles, meaning those individual bundles of those muscle fibers that are there. And when I say it this way, I mean the entire bundle itself. So the whole muscle with every bundle is enclosed class here within that epimysium. So going in a bit further, we have these that are known as fascicles. So a fascicle includes a number of different muscle fibers. So when I say it has a number of different muscle fibers, I'm saying that those individual known as fascicles, a lot of different muscle fibers, such as you would have making up maybe an extension cord. So here it's called the perimysium that separates those individuals that has each of those fascicles within itself. And then even smaller, would be class the endomysium. And the endomysium class is what separates each individual fossicule itself. So again, that is what makes up class the connective tissue covering of muscle. And just bear in mind that the figure you're seeing here has been on the test in the past and will likely be on your test class in your not so distant future. And you won't have to label everything that's there, but I'll definitely class look for you to know, of course, these connective tissue coverings of skeletal muscle. So let's continue on with skeletal muscle fibers and these terms. So this is by and large class a lot of anatomy, but do keep in mind with this anatomy class will come physiology, the function. So I'll begin where I've left off, and I left off class with thick and thin filaments. Yes, indeed. So the myofibrils, they play a fundamental role in the muscle contraction mechanism. And as I say this class, I'm coming from page 294, where it says skeletal muscle fibers. I'm here, class, in that second paragraph. They consist. So they consist of those two contractile proteins, or those two protein filaments. So those thick filaments, class, are composed of the protein myosin, as opposed to, of course, those thin filaments composed primarily, class, of that protein known as actin. So it says two other proteins will be discussed later. Well, I'll say later is now, not now and later, like the candy. Let's get to later. So with that being stated, class, I've turned the page to page 296. And on page 296, I've jumped down, class, to the last two full paragraphs, where it says thick filaments are composed of many. Thick filaments, class, it says, are composed of many. I'll give you all just a moment to get there. I believe you all should be there by now. I shall now continue. So as I've gotten to this point, what it states here is that the thick filaments, yes, they're composed of many molecules of myosin, and each of those, class, consists of those two twisted protein strands with those globular parts called heads. They project outward. So what I'm getting to here is that those myosin heads, they must attach, class, to the active sites. I said those myosin heads must attach, class, to those active sites on actin. I'll say more about that here shortly. So I'll continue now, class, to those other proteins. So those other two proteins are known, class, as troponin and tropomyosin. So the other proteins, class, are known as troponin and tropomyosin. Here we are. So if you look closely, class, troponin will be that little blue portion you see, and then tropomyosin, class, would be this, I guess you say, purple portion. So again, I say troponin, class, is here in blue, and then tropomyosin, class, is there in purple. And this, class, is what's surrounding the thin filament. This is what, class, is surrounding what is known as actin. And in the center, class, that is in a purple, are those myosin heads, and that is, class, the myosin. So now that you all know where you are, I'll continue with what I was describing, class, just moments ago. So what happens is tropomyosin, class, which is there in purple, is what blocks. And when I teach this, a lot of times I call it the blocker. I'll say again, tropomyosin, class, is the blocker. And what it does is it blocks, class, those myosin binding sites on actin. It blocks the myosin binding sites on actin, or those active sites on actin, excuse me. I'll say it one more time to say it well. Tropomyosin, class, blocks the myosin binding sites on actin, or even the active sites on actin, or however you like to put it. So, as I've stated that, what happens, class, is we have differing types of troponin. But before I get to troponin, what I'll say troponin does, class, is it's there to bind, class, with calcium. I'll say again, calcium ions, and I'm kind of foreshadowing here, from the sarcoplasmic reticulum, which stores those calcium ions. Class, please take great notes. I'll say again, troponin, class, which is there in blue, binds to calcium. The calcium class comes from the sarcoplasmic reticulum. So as the calcium ions bind to troponin class, it causes a shape change in troponin, which then class rolls away the tropomyosin from those myosin binding sites on actin. Or you could say it as being, it moves away class troponin, that blocker, from the active sites on actin, exposing those so the myosin heads class can bind to the myosin binding sites on actin and, of course, form class cross bridges. So troponin class has a three-piece, and I don't mean chicken. The three-piece class would be, of course, troponin C, or TNC, that binds to calcium. You have troponin T class, which, of course, would be TNT, thanks a lot, tropomyosin. And then, of course, we have troponin I, TNI. And I won't go into a lot of detail, class, about those three differing types at the moment, but keep in mind, class, this is needed for muscle contraction. And to get back, class, to where I left off. So I mentioned just moments ago, class, that the sarcomere, of course, is that functional unit class of muscle contraction. But what I meant to get to, class, is that sarcoplasmic reticulum. And it is that, meaning the sarcoplasmic reticulum, that stores the calcium. You must have calcium. Without calcium, class, no muscle will contract, period. And our next class are going to be transverse tubules. And when I mentioned the transverse tubule, or that triad, meaning, of course, there is, of course, one T tubule, one transverse tubule, and two, class, cisternae, and two cisternae. So with that, you see on page 297, class, that's where I am. On page 297, what happens, class, is the sarcoplasmic reticulum is here. And I'll say it has to be there because, yes, it's there to store, meaning it stores, class, the calcium ions that are needed for contraction. Well, what then happens, class, is those transverse tubules, they extend deep. And I'll use this yet again. They extend, class, deep within the muscle fibers. Those T tubules do. And the reason they do that, class, is to allow that nerve impulse to get deep within that skeletal muscle fiber. So I'll say again, the transverse tubules, the T tubules, that triad of which, meaning with the cisternae class of the sarcoplasmic reticulum, two of those, and then that one T tubule making that triad, it allows the nerve impulse, class, to get deep within that muscle fiber, class, to allow, of course, contraction, class, to ensue. It must occur. I repeat, it must occur. So there you have it. I hope you all join me, class, for those open lectures, and I would love to better explain this and answer any questions you all might have. I'll now, class, move on to what's next. And what's next, class, is what occurs, class, at the neuromuscular junction. I'll say again, what is next, class, is the neuromuscular junction. You'll find this, class, beginning on page 297. This is section 9.2, skeletal muscle contraction. Now, having said all of that, I'll say this. Class, this, of course, will be one of your short answer essays. This is what will be turned in, and this, class, is what you all will explain in the test. The events, class, occurring at the neuromuscular junction. Let us begin. If you are not well versed in what I'm going over, class, I say please reach out and I will help you. I would like to help you. And if there's anything you don't understand, you should tell me, because if you all do not tell me, I will not be able to help you, because I have to know, class, where you are for me to help you. Let's begin. The neuromuscular junction. So the neuromuscular junction, as you're seeing, class, here at the top right, this, class, is that synaptic knob, or that, I guess you could say, yes, that synaptic knob. And I'm saying that synaptic knob there, and the reason I'm saying that because that's, class, part of a motor neuron. I repeat, that's part of a motor neuron. And if you're wondering, what am I saying? Well, what I'm saying is this. No contraction, class, will ever occur unless there is first, class, a nerve impulse. Now, it may very well be that you say, a nerve impulse, yes, it must be, class, firstly, electric. And then after that has been class electric, it's then changed, meaning the messages. It's changed class into something that is called a chemical messenger. Change into a chemical. The chemical is listed there class beneath mine called acetylcholine, or listed class here called a neurotransmitter. And then the message class yet again becomes electric, meaning another nerve impulse. So I hope you're writing this down as far as what I'm getting at. No muscle class will contract unless first things first, there is a nerve impulse. So the message class is going to be firstly electric, then it's chemical, then back to being electric, and then finally class, contraction can ensue. So let's begin. And those parts class, I hope you know those parts, but if you don't know those parts, it says the motor neuron, the motor end plate, and looking very closely class, that motor end plate is just where it's there with that muscle fiber. And thereafter it mentions the synaptic cleft class, and that synaptic cleft is just that space between class, that synaptic knob and the muscle itself. That is that synaptic cleft right there. And then it says synaptic vesicles. So these class are those synaptic vesicles. And then lastly, of course, is that neurotransmitter. So at the neuromuscular junction, a number of things happen. The first thing that has to occur, class, is, of course, some form of stimulation. So after that stimulation class has occurred, as long as it reaches threshold class, then an action potential class will be elicited. Once the action potential class is elicited, class, via that motor neuron, it will then, class, arrive at the axon terminal. So if you're writing this down, which I hope that you all are, this is where we are. The action potential class has traveled from the dendrites by way of the cell body class, which contains the nucleus, and the nisso bodies. And it went down that axon hillock, and then down that long axon, and ultimately, class, made it here to that axon terminal. So it's now, class, at the neuromuscular junction. So what then happens is, as the action potential class moved down, it caused, class, there to be a change in voltage. Notice this is, class, what I call a motor neuron. So this process began when that nerve impulse, class, reached the axon terminal, and it opened, class, I repeat, it opened voltage-gated calcium ion channels. And this is in that axon membrane. So we're still, class, in the motor neuron. So it's all because of that change in voltage. So that cell, class, at rest is at an approximate negative 70 millivolts. So as the action potential class travels down, there is a change. Hence it's referred to, class, as being a voltage-gated calcium ion channel. So those voltage-gated calcium ion channels, they open as soon as it's that quick reversal, going all the way up, class, 100 millivolts to that approximate positive 30 millivolts, that the action potential. So that, class, causes those voltage-gated calcium channels to open and allow, class, calcium to rush into that motor neuron at that synaptic terminal. So at that axon terminal, what then happens, class, is that there will be synaptic vesicles to fuse, and those vesicles, class, that fuse are filled with the neurotransmitter known as acetylcholine. I'll go back and review. Because there was adequate stimulation, and I said adequate stimulation, meaning that there was a nerve impulse that at least reached threshold potential. Or at least, I'm sorry, there was a stimulus, class, that at least reached threshold potential, that then, class, caused a nerve impulse that traveled, class, by way of the dendrites, the cell body, with, of course, those nisso bodies, or the chromatophilic substance, down the axon hillock, down the axon itself, class, and ultimately to the axon terminal. That, class, caused those voltage-gated calcium channels to open, and of course, calcium entered into the axon terminal. So with that, that causes, of course, synaptic vesicles to fuse, and those synaptic vesicles, class, are filled with acetylcholine, the neurotransmitter. Thereafter, class, those vesicles that fuse, they will bind, class, to that neurolemma. They'll bind class to that cell membrane of this motor neuron, and then by way of exocytosis, class, the neurotransmitter will leave and diffuse across the synaptic cleft class to those chemically gated, I repeat, to those chemically gated sodium potassium channels. I repeat, release of acetylcholine from those synaptic vesicles causes acetylcholine class by way of exocytosis to move across the synaptic cleft and then bind to the acetylcholine receptors class on the sarcolemma, which is, of course, that cell membrane class of this skeletal muscle, opening class the chemically gated sodium potassium channels. I hope I'm going slow enough. So what that will then indeed cause, class, is it would allow more sodium class to enter and further depolarize the sarcolemma and even propagate, meaning cause that action potential class to spread in both directions, class, on that skeletal muscle fiber and within that skeletal muscle fiber. We now have the action potential class spreading, this depolarization event spreading class across this sarcolemma. So as soon as that has occurred, what will now occur, class, is the termination of the effects of acetylcholine. So acetylcholine at present, meaning right now, is still bound to its chemically gated sodium potassium channels, meaning that greater influx of sodium class is what depolarizes skeletal muscle fiber in this area. So to terminate those effects, class, it'll happen in, I would say, for this case, one of two ways, or at least for all cases, one or two ways. The first of two ways, class, is that the acetylcholine class could just diffuse away from those chemical receptors on the sarcolemma. I say again, it would diffuse away from those receptors, or it will be, class, broken down by acetylcholine esterase. I say or broken down, class, by acetylcholine esterase. Yes, that's an enzyme because it ends in ase, the suffix it is. And it will be broken down, class, into two things, two component parts. They, class, are acetic acid and choline. It will be broken down, class, into acetic acid and choline. So this, class, has been the events occurring at the neuromuscular junction. On the exam, class, you all are going to be expected to explain this. Prepare to write this well, or prepare to type this well. I'll now, class, continue through the chapter. So as we're here, I'll get to our first clinical application. And you will find this clinical application, class, on page 299, class. This is myasthenia gravis. So myasthenia gravis, class, is an autoimmune condition. The body, class, yes, is attacking itself. So in that, this autoimmune condition, class, in which the immune system attacks part of the body, it's when the part being attacked, class, is the muscular system. So grave muscular weakness, class, is in the name. So the body produces antibodies that targets receptors, class, for the neurotransmitter acetylcholine, as you all just learned about. So this is on the skeletal muscle cells at the neuromuscular junction. Junctions. So people that have myasthenia gravis have a third class of those normal acetylcholine receptors on their skeletal muscle cells. And on a whole body level, this class, it causes weakness and that the person would be easily fatigued by way of the muscles, that is. So this condition, class, affects hundreds of thousands of people worldwide. However, it's mostly, class, in women and begins around their 20s and 30s. And in men, class, they'll be affected around their 60s and 70s. So with this, most people with this disorder, with this disease, live a normal lifespan with symptoms that are controlled, class, with a combination of treatments that include drugs, class, that inhibit, look here, class, that inhibit acetylcholine esterase. I won't say the rest of which because you all know. Hence, it increases, class, levels of that neurotransmitter, or an immunosuppressant drug, such as the corticosteroids. So the next thing I'll get to, class, is the bacterium Clostridium botulinum, class. So this grows typically, class, in anaerobic environments, such as, of course, a can. Hence, you all should be careful if you ever have canned goods that have, of course, any, I guess I'll say, structural imperfections, such as being bent or dented. So if the can, class, is improperly preserved, or even as I do can, class, yes, I do can, if that's improperly preserved, so it will produce the toxin. So if the person, class, ingests this toxin, of course, the release of acetylcholine from the axon terminal at the neuromuscular junctions, class, it is prevented. Such as, and with this, the symptoms, class, of such food poisoning include nausea, vomiting, diarrhea, almost an upset sonic diarrhea. So with this, muscle weakness, class, ensues, hoarseness, and even difficulty swallowing, and eventually the inability to breathe. So the antitoxin, class, can be administered, although correcting damage, class, that's been done cannot occur. And then they also, class, use injections of small amounts in the forehead and even other areas of the face and head to treat migraine headaches and temporarily paralyze the selected facial muscles and smooth wrinkles. And I know you've heard about it, class. It's called Botox. So up next, class, is when things come together. And if you're wondering, class, what things? Things I'm referring to, class, are A, the nervous portion, that excitation portion, class, that a portion that is electric with that contraction portion, meaning you must bring the two together. So it's referred to, class, as being that excitation contraction coupling. So with excitation contraction coupling, meaning I gave you the events occurring, class, at the neuromuscular junction as being that first of two. Well, what else you all will explain on the test will be the excitation contraction coupling, and it shouldn't take too long. Let's get to it now. This begins, class, on page 298, and it continues on, class, to page 300. I'll use this figure, class, to explain it. Let us begin. So muscle contraction, class, begins, of course, thereafter, meaning immediately after those events, class, that occur at the neuromuscular junction. So I'm picking up here, class, immediately after, of course, we've had acetylcholine, class, to bind to its receptors on the sarcolemma, causing, of course, this depolarization event. So let us continue now. So what happens, class, is we have a release of calcium. I repeat, what happens now is that there is a release of calcium from the sarcoplasmic reticulum. And what happens there is that the calcium binds to troponin, as I mentioned earlier, class, which changes its shape, and then it rolls away tropomyosin, class, that formerly blocked those active sites on actin, or those myosin binding sites on actin. So now those binding sites, class, are exposed. So now what will occur, class, is that those exposed binding sites on actin will allow, class, the contraction cycle to occur. So as that contraction cycle, class, is allowed to occur, it will be all due to, class, calcium. So the calcium, of course, must reach a certain level, meaning unless there is sufficient calcium inside, there will not be contraction. So as those calcium levels rise, class, yes, we're now here. So the binding sites, class, are exposed on actin, and the events of cross-bridge cycling, class, they occur now in rapid succession. So the myosin heads, class, using, of course, that ATP, and you're not necessarily seeing it here, class, meaning it already happened. And if you're wondering what I mean is, if you look at here at number 6, class, the ATP was split, meaning it was hydrolyzed. It was split, class, and now formed ADP. Well, that, class, is what, first things first, class, broke the cross bridge, and secondly, it recocked the myosin head. Back to class where I was at number 3. So the myosin heads, class, now having, of course, been cocked earlier, will bind, class, to their binding sites on actin, forming the cross bridge, connecting, of course, myosin and actin. That is a cross bridge. So nextly, we get, class, that power stroke. I repeat, the next thing that occurs is that power stroke. So that myosin head will then, class, pull that thin filament will then pull that actin class toward that M line. I said M line, M as in Michael. It pulls it, and that class is what makes that muscle fiber slide and shorten. So ADP class and the inorganic phosphate, they are then released from myosin, and that cross bridge, of course, pulls that thin filament. The power stroke has occurred. So now we take class new. I repeat, additional ATP class binds to the myosin, and that is what class breaks the cross bridge. It breaks that connection to actin. So now that it's been broken, meaning the cross bridge, what will then occur, class, is that ATP will be split. It is used. It is hydrolyzed, which provide class the power to yet again recock that myosin head, and it's now class prepared, of course, to attach yet again class to its myosin binding site on actin. So if this, of course, acetylcholine is yet and still a class attached, if it remains class on its receptors on that sarcolemma, then of course calcium class will continue to bind to troponin, which of course will keep that tropomyosin removed from the active sites, and it will continue class in great succession, meaning, excuse me, with each contraction cycle, which I did one cycle. I hope when you test class you can do at least 2 cycles, but with each cycle class, the myosin head takes another step by attaching class to another actin site further along the thin filament, shortening class that skeletal muscle fiber. So that class has been cross-bridge cycling. And one thing that I'll do to help you with this class is that with all of this occurring, class, it had to first begin class with a nerve impulse. We're not quite there to the 10th chapter, but I'll give you an overview. So in what I'll get to here, class, is that our cells, yes, at rest, they're there, class, at an approximate negative 70 millivolts. So what happens is there is some stimulus, and that stimulus, class, may cause a bit of depolarization. It opens a few sodium ion channels. So that, class, will cause that cell to become less negative or even more positive. So maybe for that negative 70 millivolts to maybe, of course, I guess you say negative 69 or negative 68 or negative 67 and negative 66. I'll stop right there. So it begins, class, to depolarize the cell. And yes, this already, class, happened in the motor neuron, but this very same process, class, occurs in that skeletal muscle fiber. But I will say this, unless, of course, that depolarization reaches threshold potential, which is at approximate, class, negative 50 to, I guess I'll say, negative 55 millivolts, nothing will occur, meaning no action potential. So if it does, class, reach threshold, then, of course, you get the overshoot, the action potential, at that positive 30 millivolts. And then, of course, repolarization, class, ensues immediately thereafter by way of course, meaning the efflux of class, those ions. And you say, what ions? Well, it'll be the class, the efflux of those potassium ions. So as potassium channels open and the potassium ions leave, the cell then, class, becomes more and more negative, or of course more and more repolarized, being slightly, class, more negative inside and slightly more positive outside. And that's due, class, to, of course, more sodium ions, class, at rest being outside the cell, and of course more potassium ions, class, being inside the cell at rest. And that, class, has been how it occurs with both, class, the electric and of course the contraction portion of contraction cycling. And I'll do more, class, with what I just did with you in the next chapter. But just keep in mind, class, by dephosphorylation, class, of ATP into ADP, this process occurs. And it's explained, class, beginning there and continues on to page 301 in your text. And one thing that I will mention, class, that I don't want to forget is that in the absence, class, I repeat, in the absence, class, of ATP, those cross bridges, class, will ensue. I'll say again, in the absence of ATP, class, those cross bridges will remain. And if you're wondering why I say that, you just saw it here, class, here in figure 9.9 as I explained the process. Cross bridges, class, are broken using ATP, and of course cross bridges yet still are formed the very same way. So when we die, whenever that is, rigor mortis, class, will set in. And you say rigor mortis? Yes, I said rigor mortis, class, will set in. And the reason that rigor mortis, class, occurs is because of that lack of ATP class to break those cross bridges. Hence, of course, that person, or not that just that person, but hence class, we of course become stiff upon death. And that class in and of itself can be used to determine even class how long you and I class have been dead. For instance, this called rigor mortis class, it just illustrates the fact that those cross bridges will remain class unless, of course, there is ATP. So most muscles class begin to stiffen approximately 3 to 4 hours after death, and the rigidity class peaks at 12 hours, and then it just gradually dissipates class over the next 48 to 60 hours. So the cells class, of course, the dying cells, are unable class to exclude, meaning to get rid of that calcium. And that calcium influx in the muscle is what, of course, promotes formation class of the myosin cross bridges. So after breathing stops, class, we can no longer make ATP. Thanks a lot, cellular respiration. So this class is why a medical examiner can walk in to a place somewhere class on Earth and say that he or she class has been dead for X amount of hours or just a really long time based on that. So as we do relax, one thing that I did not mention earlier class is the way in which calcium, of course, has to be pumped back into the sarcoplasmic reticulum. I didn't mention it, but I should have mentioned it, so here I am mentioning it now. That calcium class is majorly important to muscle contraction, and it has to return class back to the sarcoplasmic reticulum. It has to. And I guess that is enough. Up next class, we'll get into energy sources for contraction. You find this beginning class on page 302. On page, and before I go there, I will say this, muscle fiber contraction class is outlined for you all in table 9.2. And of course, the relaxation class of a muscle fiber is outlined, the very same way. Those could help you greatly. So energy sources class for contraction is made class in a number of ways. What number of ways? 3 different ways. First things first class is by, I guess I'll call it direct phosphorylation, or at least by creatine. So we have what is known as creatine phosphate. So as you see being depicted for you there, class, is that creatine phosphate is quite abundant in muscle fibers, meaning it's up to 6 times, class, more abundant than ATP. So it can be used. So if you're wondering how it be used, class, well, creatine phosphate, that terminal phosphate class, I repeat, that terminal phosphate class is there. And it'll be used, class, to, of course, synthesize more and more ATP. So that inorganic phosphate, in the name, creatine phosphate, will then be used, class, to phosphorylate class adenosine diphosphate into class adenosine triphosphate to allow our muscles, class, to continue doing the work they do. This is especially, class, important in, well, watching, I guess you say, maybe the Olympics, which is amazing to see how those athletes can do all of those things that they do. But even, of course, as you do those things that you do, such as those short bursts of energy, even if you happen to lift weights. So the next portion will be ATP reserves. Yes, there is a small amount of ATP, class that will be used directly. And thirdly, class will be by way of cellular respiration. Cellular respiration, class, it takes time. And this is used, class, to synthesize so many more ATP molecules. And I did say so many more, and I say so many more, class, because it's approximately 36 to 38. 36 to 38. That class is so much more efficient, class, than you could ever get, class, from making, of course, only 2 ATP by that anaerobic way, or even, of course, by making a class by way of creatine phosphate. And it takes time. So this happens, of course, by way of those who may run marathons. And I did say those because I have not run a marathon yet. I did say yet. So I'll continue on, class. And before I get to oxygen debt, one thing I will mention, class, is myoglobin. You'll find myoglobin, class, just above oxygen debt on page 303. So myoglobin, class, it gives our muscle cells that reddish brown color. And I know you all have heard of hemoglobin, those heme groups. Thanks a lot, hemoglobin. It has an affinity, class, to bind with oxygen. But even class myoglobin, here of course found in our muscles, has that affinity to bind oxygen too. And in fact class, it's here for oxygen storage. So it stores oxygen class. And we have to have it because as those muscle fibers class are doing work, I did say doing work, they lose class, of course, blood because as muscles contract class, they compress those blood vessels and of course shunt that blood back to the heart by way of those veins. So with oxygen debt class, the lactic acid threshold, so this goes class from that aerobic process to that anaerobic process class of synthesizing energy. So as it happens class, meaning going from the aerobic to anaerobic, this is during strenuous activity such as lifting, I don't know, dumbbells or getting squats in, or even the bench press, which I haven't done in a number of years, I would say. So as it happens class, this is when what's done before, meaning what is by way of creatine phosphate, or even class what's done by respiration, when that cannot class synthesize the oxygen that we do need. I don't know why I mentioned class making ATP by way of creatine phosphate, but meaning when you cannot class get the oxygen we need, lactic acid is produced. So the next portion of that, I'll get down to the oxygen debt. You find oxygen debt class on page 304. So after, of course, having that lactic acid that dissociates to, of course, form lactate ions, and then, of course, they'll leave the muscle. Well, now I'll get to oxygen debt. So this is class as that lactic acid accumulates, the person develops this oxygen debt, or that excess post-exercise oxygen consumption. So it must be class repaid. So the debt class includes the amount of oxygen that the liver cells require to convert the accumulated lactic acid to glucose, as well as the muscle cells that require the resynthesis class of sufficient ATP. So this debt class also reflects that oxygen requirement to restore blood and tissue oxygen levels to those pre-exercise levels. So if you ever class been out exercising, and of course you felt that need, this is why, because of that debt of oxygen class from you all, of course, using it up. Not to say that you had no oxygen left, but of course it not being class at that level that it needs to be. And the next thing I'll get to is, I guess I'll go down to muscle fatigue. So muscle fatigue, as it occurs, is just when your muscles class are not able to contract. So meaning when that muscle is exercised and exercised and exercised for that prolonged period of time, and it may just be mere seconds in the event that you're training, I guess I'll say, with free weights. Well, as it happens, class, the muscles may lose their ability to contract, and it may be because of a decrease in blood flow, ion imbalances, or just that repeated stimulation from doing all those reps, or those repetitions rather. And it could also have been, class, from the physiological loss of the desire to continue to exercise, which I hope hadn't been the case, or will be the case ever. So from here, class, I've already mentioned the way in which, well, let's go to muscle cramps. So this is nothing more, class, than meaning changes in electrolyte concentration, class, in our extracellular fluids in that area. We must have, class, the appropriate electrolytes in the body. I haven't mentioned a number of lab values to you all yet. However, there's a certain number. When the number, class, falls, we get this sustained involuntary contraction that you all may know as a cramp, or others of which may call this class called Charlie horse, I think it is. Up next, I was going to mention moments ago, is heat production, class. Of course, class, our muscles produce copious amounts of heat. So next up, I'll get down to muscular responses. Hello, buddy. How you doing? How you doing?
Can I come?
Yes, I'm just making a video. It's about muscle. The video is about muscle. I'm almost done, too. You ready?
Yes.
I'll continue. So first things first is that threshold stimulus. I've mentioned this a number of times, meaning there won't be a muscle class to contract unless that stimulus class has reached threshold.
Oh.
Yes, threshold. Can you say threshold?
Oh, threshold.
Yeah, say it again. Threshold.
Threshold.
Okay. So upon threshold stimulus being reached, class, that will then allow just one more sodium ion to enter and then surpass that threshold stimulus, or excuse me, that threshold level to, of course, elicit a nerve impulse. So once that nerve impulse, class, has been elicited, we then, class, get the action potential. And I've mentioned this already. As that happens, class, meaning the threshold stimulus. Yes, go ahead. As that threshold stimulus has been reached, well, contraction, class, will of course then ensue. So that nerve impulse, class, then goes deep into that skeletal muscle by way of course, the T tubules, the triad I mentioned earlier, which then, class, causes the release of the calcium ions from that sarcoplasmic reticulum, and then, of course, thereafter, class, ultimately getting the cross bridge formation by way of the, of course, calcium influx, and then binding, class, to troponin to remove tropomyosin, class, and I'll just stop there because I just explained it. So it's all because of that action potential, class, from that motor neuron, releasing acetylcholine to produce, class, every one of these effects. So first things first, I said it earlier, electric, chemical, and then of course electric again, and finally, class, there is that muscle contraction. So now I'll get to recording, class, of a muscle twitch. This figure won't be in the test, but I like the figure because this figure, class, shows those events. First things first, class, says simulation. If you look closely, class, there is a latent period. The latent period, class, is quite important. Meaning, even though, class, is approximately 2 milliseconds, the latent period is. Everything I just taught you, class, is what's occurring in that latent period. You're saying, what do you mean? Well, electric, chemical, electric, and then, class, you see the period, class, of contraction. So just keep in mind, class, that there must be some latent period thereafter, because it does not, class, occur automatically or even concurrently. So up next, we'll get to isotonic versus concentric contractions, or at least eccentric contractions. So the isotonic contraction, class, this occurs because the muscle actually shortens. I repeat, the muscle shortens, known as the isotonic contraction, or you could say the concentric contraction. And it's all because, of course, that equal force equals that change in length. And in other words, the muscle contracts with that force greater than the resistance being applied, and of course, you can now curl. So the other type of isotonic contraction is known as that eccentric contraction that contrasts the concentric contraction. So this eccentric contraction, class, happens when that muscle contracts with force less than the resistance and then lengthens. So this happens, class, as you, I guess I'll say, finish your curl by, as some may say, get the negative, as they say in the weight room. And then lastly, class, will be the isometric contraction. And the isometric contraction occurs, class, when the muscle contracts but does not change in length. Next up will be fast and slow twitch muscle fibers. I'll say it briefly and shortly that this is the reason, class, that sports are what they are today. Slow twitch muscle fibers, class, are just as that. So they can, of course, function for long periods of time without fatigue. In other words, the marathon runner. Fast twitch glycolytic fibers, class, in their case, can, of course, contract so rapidly, but then again, they are quite, class, susceptible to fatigue. And then, of course, the type 3 fast twitch fatigue resistant fibers can also, class, contract, and then, of course, contract, and then, of course, contract. So the muscles, of course, they have an array of these. And, of course, with this is the reason, class, that someone can maybe hit a ball so far or run so fast. Muscle tone is just that simple continuous state of partial contraction in your resting muscles. So this occurs, class, from, I guess I'll say, working out. Now on to clinical application 9.2, use and disuse of skeletal muscles. I'll keep this easy. Atrophy class occurs from disuse. Once I had a fracture, a Lisfranc fracture, and from that Lisfranc fracture, I had surgery. Muscle atrophy ensued. And of course, it's just as that. I was unable to walk, I had crutches, and I could not walk at all. I had to use crutches to get around, if you can call that walking with crutches, as opposed to class hypertrophy. So hypertrophy class occurs by way of forceful exercise, such as weightlifting. And with this class, the muscle can get larger and larger and larger. So that just by forcefully exercising. And I'll put it this way, just make sure class that you all are exercising. Exercise class is good for you, but I'm no doctor, so make sure you consult a physician, your physician class before beginning any exercise regimen. I don't recommend you beginning it without that. So now on to smooth muscle class. This is on page 308 of the textbook. And with smooth muscle, it's markedly different class from skeletal muscle. And I did say markedly different. So smooth muscle class can be referred to as being multi-unit smooth muscle, or even, of course, visceral smooth muscle. But they have a single centrally located nucleus. Those cells class are, as I mentioned earlier, elongated cells, and they're thinner. And they lack striations, of course, and that's why it's known as being smooth muscle. So with this class, visceral smooth muscle, I'll refer to it class. And the reason I say this is because visceral smooth muscle class responds as a single unit. So when one cell is stimulated class, the impulse is conducted over the surface to excite adjacent cells, and of course, they in turn stimulate other cells too. So it's because of this that some visceral smooth muscle also exhibits or displays rhythmicity. So these two features of smooth muscle being conduction of impulses class from cell to cell and rhythmicity are largely class responsible for the wave-like motion known as peristalsis. And you may have heard of peristalsis before, and I'm referring class to peristalsis of the gut. This class is what moves foodstuffs through the body. I say again, it is peristalsis class that moves foodstuffs through the body. And if you're still wondering what do I mean, well, what I mean by saying this is drink a sip of water. You may have drank that sip of water, but as soon as you are finished class drinking that sip of water, the process class continues on without anything class coming from you. And I say class, that is the essence of smooth muscle. That propulsive action that happens to, I guess I'll say, it squeezes things class through the internal organs pathway. And the same thing occurs class as you begin the process of digestion. I mean, this in and of itself class is what then moves everything class through the digestive system. So it's also class peristalsis that, of course, in the ureters, that moves urine class from your kidneys to the urinary bladder. And I guess I'll just keep going since I've gotten started. So it's peristalsis class that consists of those alternating contractions and relaxations of the circular and longitudinal layers of smooth muscle. So this is what mixes contents of your food class and pushes them, of course, along the length of your intestines, moving masses of digested food to help mix them class with digestive fluids. And then, of course, it's that visceral smooth muscle, as I just mentioned. That is, of course, that more common type of smooth muscle found in the walls of your hollow organs, such as the stomach, the uterus, bladder, urinary bladder, as I mentioned, and the intestines. So let's get further into smooth muscle here. So smooth muscles, it has what are known as varicosities. And these are just those bulbous swellings. So those varicosities class are what release neurotransmitter into the synaptic cleft. And one thing that is different class is that There are no T-tubules in smooth muscle. However, the sarcolemma class has multiple caveolae, or pouch-like end foldings. As I mentioned, those things known as caveolae, meaning those end foldings, they contain class large numbers of calcium channels. So when calcium channels class and the caveolae are open, calcium comes inside, meaning there's an influx of calcium. And with that, the calcium enters the calcium channels directly from the extracellular space. So to reiterate what I was saying, class, there is no troponin complex. As far as smooth muscle contraction occurs, class, smooth muscle cell fibers, in the stomach and small intestine, class, they are pacemaker cells. And I'm saying that because it's that they are setting the pace of contraction, class, for the entire muscle sheet. And one thing I will also mention, class, as far as smooth muscle, is that calcium activates the myosin by interacting, class, with the regulatory molecule known as calmodulin. So smooth muscle has calmodulin, which of course is a regulatory molecule. So from there, let's get to regulation, class, of contraction. So it's regulated, class, by, of course, neurons, so it can be regulated neurally. And with neuroregulation, it will happen as being that effect of a specific neurotransmitter on a smooth muscle cell. It depends, class, I would write this down. It depends, class, on the receptor molecules on that cell's sarcolemma. I have an example of that. When acetylcholine, class, binds to acetylcholine receptors on acetylcholine receptors on smooth muscle in the bronchioles, class, that's found, class, as being those small air passages of the lungs. The response, class, is strong contraction that narrows the bronchioles. And that's when acetylcholine, class, binds to smooth muscle receptors in the bronchioles. However, when norepinephrine, class, which is released by a different type of autonomic nerve fiber, binds to the norepinephrine receptors on that same smooth muscle fiber cell, the effect, class, is inhibitory, i.e., the muscle relaxes, which dilates the bronchioles. But keep in mind, class, when norepinephrine, class, binds to smooth muscle fiber in the walls, I repeat, in the walls of blood vessels, it stimulates the smooth muscle cells to contract and constrict the vessel. I'm saying this, class, because it's pretty important to know the receptor that predominates, class. If you just give someone, class, norepinephrine, or just give them, class, that acetylcholine, it could potentially, class, kill him or her. The effect, class, is receptor dependent. I'll continue on now, class, to hormonal and local chemical factors. Briefly, class, here I'll mention that it's going to be several chemical factors that cause smooth muscles to contract or relax without an action potential, class, by, of course, enhancing or inhibiting, class, calcium entry into the sarcoplasm. Certain hormones, histamine, excess CO2, carbon dioxide, that is, low pH, or even a lack of oxygen, class. So all of this, class, affects whether or not smooth muscle contract, and is even, class, some special features. So up to a point, class, when skeletal muscle is stretched, it responds with more vigorous contractions. However, class, stretching of smooth muscle also provokes contraction, which automatically moves substance, class, along that internal tract. I mentioned the hormone oxytocin, class. It also stimulates smooth muscle in the wall of the uterus to contract. That was one hormone that I needed to mention. And even stretch, class, below that urinary bladder. So just keep in mind, class, that unlike skeletal muscle, smooth muscle cells can change length without changing tautness. And this is what enables, class, smooth muscle in the stomach and intestinal walls to stretch as these organs feel yet maintain constant pressure inside the organs. And what I'll get to nextly will be cardiac muscle. So with cardiac muscle here, class, what I'll say about it will be brief because in Anatomy 2 class, Human Anatomy and Physiology 2, you all will have chapters, and yeah, that's plural. You all will have chapters on the cardiovascular system and cardiac muscle. So earlier stated class is found only in the heart, composed of striated cells joined end to end, forming interconnected, branching, three-dimensional networks with a single nucleus class. So the opposite ends of cardiac muscle cells are connected class by intercalated discs, and these bands are complex intercellular junctions that include components of desmosomes and gap junctions. That class was also mentioned to you all back in the prior chapter, being chapter 5. Intercalated discs also help join cells and transfer the force of contraction class from cell to cell and allow ions to diffuse between cells. Ions class are majorly important in the cardiovascular system. So the whole structure class contracts as a unit or a functional syncytium, I'll say. That is class that the network responds to stimulation class in that all or none manner. It won't be that part of it contracts. It will be it all contracts or it all will not contract. Hence class is that syncytium. Find this class at the bottom right hand side of page 309. And lastly, I'll say that cardiac muscle class is self-exciting and rhythmic. So a pattern of contraction and relaxation class repeats, generating class the rhythmic contraction of the heart. So cardiac muscle becomes class non-responsive, or at least refractory, after stimulation class until the contraction ends. Hence class, sustained contractions, as I mentioned a little bit about, or tetanic contractions, as it occurs class in skeletal muscle, do not occur in the heart. So class, this has been chapter 9. And this chapter class is quite long, but as I stop, I'll recommend you all stop. And of course, I say start back when you begin learning class, the muscles for the test. But otherwise, class, this entire chapter will not be on this lecture test. Stop right there where I stopped. As far as lifespan changes, class, as they occur, as we enter into our 40s, which I have not yet, thankfully, the signs of aging class in the muscular system begin to appear, even though the person class is still active. So just keep in mind, class, activity is good for you and good for me. If you all, class, have any specific questions or even general questions or any questions at all, I recommend you ask. Please join me for those open lectures. Please join me for office hours via phone class, via face-to-face, via maybe not face-to-face today or next month even. But just meaning contact me so I can help you. I know this isn't easy, but it's definitely, class, what you all can do. So please take care of yourselves, class, study well, and prepare for the test. This has been your instructor, Scholar Huff.